All right, welcome everyone. Step right up to the chalkboard for today's masterclass. I'm incredibly excited to share this explainer with you because honestly, what we're looking at today feels like absolute magic, but it's actually pure, rigorous science. We're going to explore how a tiny, almost microscopic speck of a plant, like just a fragment of a leaf or even a single cell, can literally be guided to grow into an entire living, breathing forest. So grab your notebooks because this is going to be a fascinating ride. Okay, let's dive into this. It's a completely mind-bending biological premise chalked right up here on the board. Can one single plant cell really grow an entire forest? In the animal kingdom, if you isolate a skin cell, well, it's just going to stay a skin cell. But plants, they've got this secret superpower hidden deep in their biology that lets them play by an entirely different set of rules. And that superpower is called totipotency. This is the magical biological foundation that makes plant tissue culture actually work. To put that into perspective, in animal biology, the only fully totipotent cell is a zygote, the very first cell formed by fertilization. But in plants, almost any mature differentiated cell, as long as it has its original intact genome, can theoretically be completely reprogrammed. It can essentially de-differentiate, revert back to a blank slate state, and then redifferentiate to form a completely new, perfectly functional plant. It is, quite literally, biological time travel. Now, what's really interesting here is seeing this concept actually play out under the microscope. What we're looking at is an actual, immature embryo of a pine tree being cultured in vitro. It's been carefully isolated from its normal environment, placed on this highly controlled artificial nutrient medium, and then triggered to grow. We are literally watching life being coaxed into existence from a tiny little fragment right inside a laboratory. So let's move on to section 1, the culturing process, taking this from the lab to the land. To understand exactly how we harness this superpower, we really need to map out the recipe used to clone these plants. Just imagine a chalk line connecting a healthy mother plant sitting in a greenhouse, all the way to a massively multiplied crop out in an agricultural field. This whole lab-to-land journey happens in 5 distinct stages. We've got stage 0, which is selecting our stock plant. Stage 1 is establishing an aseptic culture. Stage 2 is where the real magic of multiplication happens. Stage 3 induces rooting. And finally, stage 4 is acclimatization, where the plant gets readied for the harsh real world. Let's look a bit closer at the very beginning. We start by taking an explant, basically just a tiny piece of a healthy, disease-free mother plant. But here is the absolutely critical part. Microbial contamination is the single biggest enemy of plant tissue culture. Since the nutrient jelly we use is absolutely packed with sugars, bacteria and fungi will take over instantly if they get even half a chance. So, the explant goes through this incredibly intense surface sterilization. It's washed with alcohol and often treated with some pretty heavy-duty sterilants, like a 1% mercury chloride solution or bleach. And all of this is performed exclusively inside a super sterile environment called a laminar air hood. And this brilliantly illustrates that exact cycle. We start with a single node explant from a greenhouse plant. It's placed on a medium, it forms this massive tissue called callus, and then shoot buds start to initiate. Those shoots get elongated, they're rooted in a completely new medium, and finally, that fully formed little plantlet is sent right back to the greenhouse. It really is this perfect, continuous loop of biotechnology. Now, during the multiplication and rooting stages, things start growing exponentially. We place the explant into a nutrient-rich base, like the famous MS medium, which is named after the scientists Morishige and Skog. It sits in a strictly controlled growth room at around 25 degrees Celsius. But, it doesn't just sit there. Lab technicians actually have to constantly slice up these growing microshoots and subculture them into fresh nutrient jelly every 4 to 6 weeks. If they don't do that, the plants will completely deplete their nutrients and actually start producing toxic metabolites. Once we finally have enough shoots, we transfer them over to a specialized rooting medium. Right here we have an actual jar containing an in vitro clone of a sugar maple. Notice all that heavy condensation on the glass? That is because the environment inside that jar is sitting at nearly 100% humidity. You can also spot the delicate little roots forming down in the nutrient medium. This plantlet is perfectly happy. It's being completely pampered with all the sugars and nutrients it could ever need, with zero environmental stress. But as you might guess, a pampered lab plant wouldn't survive a single day out in the real world. And that brings us to step 4, hardening, or acclimatization. This is the final crucial test. We have to gradually transition this super fragile plantlet from its 100% humidity, perfect temperature jar out into normal, harsh field conditions. So we slowly lower the humidity and dial up the light intensity, essentially teaching the plant how to photosynthesize completely on its own and survive without its laboratory life support system. All right, moving on to section 2, the hormonal seesaw. So, how do scientists actually tell the plant tissue whether to grow a leaf or grow a root? Well, I want you to imagine drawing a seesaw right here on our chalkboard. It is this incredibly delicate, brilliant balancing act involving plant growth regulators, specifically hormones called auxins and cytokinins. When scientists tip the hormonal scale one way, adding high auxin and low cytokinin, the plant responds by sprouting roots. But if they tip the scale the exact opposite way, low auxin and high cytokinin, leaves and shoots start to emerge. By just tweaking the chemical recipe in the jelly, we can literally command the cells to build exactly what we want. And because of this amazing flexibility, there's an unbelievable variety of explants we can start with. We can culture seeds and embryos to bypass normal dormancy. We can culture the meristem, which is the very tip of the shoot, because it's actually naturally virus-free. We can culture whole organs, like leaves. We can even do something called protoplast culture, where we use enzymes to literally strip the rigid cell wall right off a plant cell. This creates a naked cell that can be used for genetic engineering or even fusing two completely different species together. It's wild stuff.
That brings us to section 3, 2 paths of regeneration.
Once we have our tissue in the jar, the plant has to figure out how to rebuild itself down one of 2 distinct biological highways. But before picking a path, the tissue often forms something called a callus. A callus is basically an unspecialized, totally unorganized mass of dedifferentiated cells. Think of it as a blank slate intermediate blob of tissue. The plant has essentially melted down its specialized cells into this raw building material, and now it's just sitting there waiting for the hormonal signals to tell it what shape to take next. Let's move to the microscope to see how this builds. These undifferentiated cell cultures just look like a bunch of loosely arranged parenchyma cells. They aren't leaves yet, they aren't roots, they are just pure proliferating potential. And from this unorganized stage, we can guide the cells down one of our 2 regenerative highways. So the crucial point here is the difference between these 2 highways. The first is organogenesis. This is unipolar development, where you prompt the tissue to sprout just a shoot, or just a root, which you then have to guide through the rest of the steps. But the second one, somatic embryogenesis. This is incredibly cool. It's bipolar development. That means a regular, non-sexual cell completely skips that unipolar step and directly forms a complete, artificial, seed-like embryo that has both a shoot tip and a root tip at the exact same time. And finally, let's wrap up with section 4, why this matters, the big picture. Let's zoom out from the microscope to the macro level. It's really fun to look at test tubes and cellular blobs, but why does all of this meticulous lab work actually matter to us? Well, because this chalkboard biology lesson is actively saving our food supply and our global ecosystems. Think about it. By the year 2050, we need to feed a projected 9.9 billion people on this planet. Traditional farming just cannot scale fast enough. But plant tissue culture, it allows us to rapidly multiply millions of disease-free, climate-resilient crops in a fraction of the normal time. It allows us to rescue rare and endangered plant species right from the brink of extinction. And it even lets us mass-produce absolutely vital medicinal compounds and pharmaceuticals right in liquid culture vats. So, I want to leave you with this provocative thought as we wrap up today's explainer. The next time you eat a piece of fruit, or admire a beautiful orchid, or even take a plant-based medicine, I want you to remember the incredible, invisible science that might have brought it into existence. How will this totipotent biotechnology continue to reshape the future of the very food on your table? It's pretty amazing to think about. Thanks for joining me at the chalkboard today, and as always, keep learning.